In this part, we'll be discussing on the evidences from the experimental studies. Experimental studies, in the sense, the whoever was involved here, they are going to set some conditions like light, temperature, and nutrients. They are going to observe how these pteridophytes are going to behave in these different conditions, and that led to formation of heterospory. Now they have chosen Selaginella and Marsilia for this experimental studies. They say on the basis of the experimental studies on Selaginella and Marsilia, it has been observed that heterospory originated due to nutritional factors. We have seen this in our previous first slide only, I guess so, in first session, in fact, showing a diagram where microsporangia led to the formation of megaspore, and the megaspore evolved into a seed. That was because of why these megaspores developed by consuming the more nutrient which was available when the degeneration of the microspore took place, and leading to formation of one megaspore. Again, that is the same factor, that is a nutritional factor. What they did, it was like observed that if the photosynthetic activity, photosynthesis in the sense they are going to produce the food, photosynthetic activity of the Selaginella was slowed down. How? By keeping it in a low intensity light. Low light was supplied to Selaginella, so obviously the photosynthetic activity is reduced. Photosynthetic activity is reduced in the sense there will be low food available, low nutrition available, small amount of nutrition was there. Then what it led to? It led to development of the microsporangia. Due to low photosynthetic activity, nutrition become a limiting factor, only limited nutrients were available, and the spores could not grow in size, because for the spores to grow in size they require less number of spores and more amount of food. Here the food itself is less in less, that it is a food is a limiting factor. That's the reason spores could not grow in size. Thus under such condition only microspores were produced. So nutritional will be utmost factor for the formation of heterospory. Similar experiments were performed on Marsilia by Shatak in 1910. What he did, he took variable conditions like light, temperature, and nutrient. He found that that in plants growing in favorable condition, favorable in the sense everything is fine, light is abundant, temperature is abundant, and nutrient condition is also fine. In that condition the microsporangium contains what? Aborted microspore. That is, some of the microsporangia, microspores present in a microsporangium are going to degenerate, showing a microspore abortion. So whatever small amount of spores are left, they are going to have sufficient amount of nutrient available, and they are going to use it and increase in their size. The same thing happened. The microspores were aborted, and whatever spores left, they were 16 times larger than the original size. 16 times larger in the sense the size of the spore has been increased, and the larger spore is nothing but a megaspore. And in that extreme cases of abortion, only in extreme cases what happens, abortion only a single spore survived, and it looked like a megaspore and showed all the structural features of megaspore. So the different conditions have led to know how exactly the heterospory evolved. So this was a beautiful experimental studies done on Selaginella and Marsilia by Shatak and all. Thank you.